Now we will see chemiosmotic hypothesis which was given by Mitchell. The Mitchell, why he has given and what is the importance of this one? He got Nobel Prize, remember it. Means how much important it is. That much of hypothesis it is. What it shows, it explains how the ATP will be synthesized in the mitochondria and chloroplast. It explains. And what it is, and this is the ATP synthase linkage with the development of proton gradient. ATP synthase means ATP, it will prepare the synthase, will do the catalyze the ATP formation. It will be worked under the proton gradient across the membrane. What is this? Is the textbook only has written. But the thing is, whatever the protons are developed, the concentration is developed in the membrane one side, will travel to the other side by the ATP synthase formation. That I will explain it. Just wait. Now the thing is, where it will be taken? It will be taken in the chloroplast of the chloroplast thylakoid and intermembranal space of the mitochondria. Mitochondria is having two layers. That is the outer layer and inner layer. In between the whatever the space is there, that is called intermembranal space. Yeah, though in between these places, whatever the proton gradient will be developed, and they will pass, the gradient will be released from higher to low through a proper mechanism that will be explained, and that mechanism will explain how the energy will come to prepare the ATP. This, if he says like this, you cannot now understand. The thing is, this thylakoid, how it is happening, first I will explain. And later on, in the textbook orientation, I will give. Now, first thing, this is the thylakoid membrane. But two layers I shown, but it is one layer only. Thylakoid membrane entire. Any membrane which is present in the protoplasm is works as a plasma membrane. Plasma membrane only. Now we go to the plasma membrane. In the plasma membrane, what you read? It is a biphospholipid layer and integral protein, peripheral protein, and its function all will be applied for this membrane. Remember, this is most important. So, in this membrane, light harvesting systems are existing. Light harvesting system, the photosystem 2 and photosystem 1. They used to present in this. And they will do, what they will do? They will do the non-cyclic. Now we will see. In the non-cyclic, what are the product? ATP and NADPH. And in the membrane, how the ATP and NADPH it will produce, how it will helpful, that we are going to explain. Now you see, in the membrane, there will be the P680, that is the photosystem 2. Used to present. The photosystem 2 used to receive the light and excited, and it will send out one of the chlorophyll molecule will be excited, and it will send out the electrons. The electrons will be received by the electron acceptor. In the jet scheme also you read, and then immediately it will give those electrons to the plastoquinone, PQ, which is present in the membrane. And when the plastoquinone is receiving electron from the photosystem 2, the plastoquinone already it will have the one proton from the stroma side. Plastoquinone is having the capacity to take proton from the stroma side, and from this one electron came The electron came from the photosystem 2 and it will be captured by the electron acceptor, primary electron acceptor, that we used to call it phosphatin. Okay, it's all right. Actually, the primary electron acceptor in the textbook he has given. And the primary electron acceptor will give the electron to the electron carrier, plastoquinone. And plastoquinone is not only the carrier of the electron. It will take one proton from the stroma, and that along with the electron, the proton also will move in the plastoquinone. This is important point. Why? What is the electron transport with the proton? It is important or what? Now you see, electron 1837 electron weight. 1837 electrons weight one proton. One proton is equal to 1837, means how many times it is. That much big size, the weight will be moved with the energy, electron energy, which came from the excitement of the photosystem 2. Means here you should understand. Everyone knows in the chemiosmotic hypothesis, it required proton pump. He has given membrane, proton pump, proton gradient, ATP synthase, electron energy, all the things. That much weight proton will be pumped from the stroma to the lumen, the thyroid membrane, whatever place is there, that we used to call it a lumen inside. So from stroma to lumen, it is pumping with the help of the kinetic energy of the whatever the electron is having. That how much kinetic energy it is? There is a lot of kinetic energy, because the excited electron means it is coming with the full force. That force is enough to lift 1837 more weight proton from the outside to inside. This is most important. Proton pump means that the plastoquinone will do this work. Plastoquinone will receive the electron from the primary electron acceptor. By the same time, it will take one proton and move this proton and electron, and comes within the membrane. It will reaches to the cytochrome B6F. Cytochrome B6F, what it will do? The plastoquinone is coming along with the electron and proton. Now the electron will be sent to the cytochrome B6. Means cytochrome B6F is having more reducing power. So the electron will be attracted by them, by the cytochrome B6F, and proton will be sent into the lumen. Proton will be sent to the lumen. This means who is separating plastoquinone from the proton and electron? That is the cytochrome B6F. This is little bit. And now the proton is carried from where to where with the help of the electron energy, kinetic energy, from the stroma to the lumen. Now here cytochrome B6F, whatever is there, it is taken the electron and send the proton inside. What is the work of the B6F? We will ask. That is separation of the electron and proton, which came from the plastoquinone. Where they send? Proton will be sent into the lumen, and the electron will be sent to the another electron carrier, which is present inside of the membrane. Remember, inside of the membrane. And here primary electron acceptor outside of the membrane. We will ask how many are inside and how many are outside also. He will ask for the time telling particularly. So inside of the membrane, one P plastoquinone molecule is to present. That is a copper containing protein. It will, the cytochrome B6F will pass the electron to the plastocyanine, and the plastocyanine will send this electron to the photosystem 1 P700. By that time, the time when the electron reaches here, the light will fall on the P7 and PS1. And this also, the reaction center will be excited and will send out one electron with high speed. That kinetic energy it will have. That energy will be, at that type of energetic electron will be received by the ferridoxin sulfur complex, iron sulfur complex, ferridoxin, which is present outside the membrane. PS1 is present, PS1 is present in the membrane, PS2 also present in the membrane, cytochrome B6 also in the membrane, plastoquinone is also in the membrane, only plastocyanin is inside the membrane. Now, the ferridoxin will give the electron to the FNR, ferridoxin NADP reductase enzyme. This electron, by that time the ferridoxin NADP reductase used to take the NADP plus one proton from the stroma. And now the stroma will provide one proton here. This electron will come, the hydrogen, it will become H proton will become hydrogen, and it will be added to the NADP and it will become NADPH. So who is preparing? Photosystem 1 is preparing the NADP to NADPH by taking the electron. By photosystem 1, the electron came and reached the FNR, and FNR given the electron to the proton which is with the NADP in the stroma side, and this hydrogen will become hydrogen atom and it will be attached to the NADP and NADPH will form. Now what you learn? Proton is pumped from the stroma to lumen. In this way, in the stroma side, proton number is decreasing, and proton is taken with, and this electron is taking the proton and electron, it will become the hydrogen. So in this way also proton is decreasing. So in the two ways, from the stroma side, protons are decreasing, and one way the proton is increasing by pumping method. The energy of the electron which came from the photosystem 2. Now, in this way, proton is pumped and proton concentration developed in the lumen. Now you see here, the thing is, in the protons, that electron or whatever the electron has gone from the photosystem 2 will be filled by the potassium of the water. That already I discussed splitting of the water in the last video. Now the water molecule will be splitted into the electron, proton and oxygen. And this in the splitting, whenever it is splitting, it is associated with photosystem 2. And this complex is present inside the membrane, remember it, inside the membrane. Now, that from that one electron will go and fill the reaction center of photosystem 2, and protons and oxygen are present. Proton and oxygen are present in the lumen. Now, the proton is, this proton came from the water. So, why I am telling specifically, this is the generated proton and this is the pumped proton. In the two ways, proton concentration is increasing in the lumen, generated from the water and pumped from the stroma to this one by the electron energy. Now you see, by the two ways the proton concentration is developing. Why proton concentration developing? What happened? The proton concentration is developing in the lumen in the two ways, and this is given high electrochemical potential gradient also here. Means here the gradient, whatever the potential, that whatever the proton concentration is increasing, that is high electrochemical. Means this is having the chemicals, ions, the chemicals of ions means protons. the protons are increasing inside the lumen. We'll keep it aside. Now the oxygen, what will happen to the oxygen? Oxygen will be diffused from the membrane. Why oxygen is diffusing, not a proton? For that only I told in the starting. It is a thylakoid membrane, nothing. It is having the characteristics of the plasma membrane. Plasma membrane will allow the that is, it will allow the neutral particles by the diffusion. Neutral particles can be diffused through the membrane of the plasma membrane. Charged particles cannot be. That you go and see the characteristics of plasma membrane. So now for that one I told particularly. So the oxygen will become O2. Means a single atom will be the charged, but it will become O2, and which is the neutral, and it will be diffused from the thylakoid membrane. But proton cannot. Proton is the positive charge. These are the phospholipids. This phosphate head is to present towards outside in the membrane. So it is a charge, water charged particles it won't allow. So protons will be inside only. So it is increasing. The concentration of the protons are increasing inside the lumen. Now the concentration of increasing by the lumen of the thylakoid and decreasing in the stroma by pumping one and adding one to the ATP. Now this, whenever it is increasing, they will try, because of the concentration more, outside stroma it is less. It will give a lot of pressure on the membrane. For that will be released by the ATPase enzyme. What is this now? What I tell you? ATPase enzyme is a protein. This protein will have the two parts. That is F0 and F1. F0 particle embedded inside the membrane, here given the book also, and forms a channel, and it is called as facilitated diffusion. What is this facilitated diffusion? Means here, when the charged particles are not allowed from the membrane, it will be facilitated by a channel from the protein. That only integral protein we studied in the plasma membrane. This is an integral protein. CF0 is also an integral protein which is having a channel, means hole. From that one, protons will come out with high force. Now one more thing you have to remember. The proton concentration is increased, for that one pressure is developed. And one more angle also I am telling you, remember, the protons are positively charged. There will be a repulsion between the positive charge. So that repelling energy and concentration energy both will be work, that it will come with full force from the lumen to the stroma through the channel of F0. I already told that ATPase enzyme will have the two fractions. One is the F0. CF0 means the channel F0. And it will be attached one more particle of the ATPase enzyme, that is the F1. So when it is coming with force through the channel which is present in the CF0 and present in the membrane, the F1 particle used to present outside the membrane, and it is a movable one, round circular and movable. Why I am telling that one? When the force, the protons are coming from the lumen to stroma, it will turns the F1 particle like this, and whenever it is turning, the ADP plus phosphate will become ATP. Means what? Here the thing is, The active sites of the enzyme used to present where, he will ask, tell me. Active sites of the enzyme used to present on the F1. What is the active site of the enzyme? Go and read the enzymes. In the enzyme, active site will work with some energy required. There ATP will be. But here, the mechanical energy which is developed by the force of the protons through the F0, the force will give the mechanical energy to rotate the F1. F1 will have the active sites. In that one already ADP and phosphate used to present, and that will be pressed. Then ATP will be prepared. This is important. If the protons are not there, other molecules, it may be not that much force. Protons are having the repulsion as their positive charge, remember it. So in this way, the breakdown of the proton gradient from the lumen to the stroma through the CF0, F1 will rotate and forms the ADP plus phosphate to ATP as given in the book. So here I am telling one more point, the repulsion energy also will be used for the sending the protons at the high speed. And next, the thing is, proton development, that is how it is happening because of the proton gradient which developed from the lumen to the stroma. So that only is telling. Now, come to the textbook. Here, thylakoid, okay, it is developed. Then what about the mitochondria? Mitochondria will be, the gradient will be developed in between the outer and intermembranous space. That you will read perfectly in the respiration. Now, this is the formation of the ATP through this one. Now, the thing is, in the textbook he has given proton gradient, how it will be developed in the number 1 heading. By the splitting of the water inside, it is increasing, generated proton. And next one is the pumped proton from the stroma to lumen. In the two ways, the concentration of the protons are developed in the lumen. So first he has given water splitting used to give rise the protons in the lumen, and it will become cause of the proton concentration increasing in the lumen he has given. Second point he has given, the electron which used to come from the photosystem 1 used to go received by the primary electron acceptor, and primary electron acceptor will give this electron to the plastoquinone. And plastoquinone is an electron carrier, not only electron carrier, it is also carries one proton which was taken from the stroma, that also I discussed. And it will release the proton in the lumen and send the electron to through the electron passage, electron transports. He has not mentioned in the text about the cytochrome B6F. Here that is the problem. He has not told about the plastoquinone, not told about the cytochrome, not told about the plastocyanin, and he has given all the names in the diagram. Who will take the response? The teacher will take the responsibility. Now I am taking. How they are, what they are. But he will ask in the NEET, what are they, what it will, how it will do, all the things he will ask. For that only I am telling, listen carefully, twice or thrice, and try to understand. If you are getting doubt, call me, I will tell you. Now the electron transport, what it will send in the plastocyanin, and plastocyanin to photosystem 1, and it will happen this I explain. Now here in the third place he has given one more point. How you will say the protons are developed, increased its concentration in the lumen means by pH. Protons are developing, pH will be fall, and OH are developing, then it will be this will be that is the called as the alkali. Now protons are developed, then it is the SDT, below 700 pH. So acidic nature they measured in the lumen. and they confirmed the chemiosmotic hypothesis. How it is happening? By the breakdown of the proton gradient from the lumen to the stroma. These are the questions. So here, primary electron, now you see protons gradient develop in the lumen by the H2O splitting and pumping of the proton from the stroma to lumen, already discussed. That is primary electron acceptor to plastophenone with the one proton also carrier and cytochrome, and then photosystem 1 it has gone. ATP synthase F1, F0 and F1 it is having, and it will be facilitated diffusion will be provided. This is the PEP, primary electron acceptor to plastophenone, which is the carrier of the proton also, and carrier it will come to the cytochrome, through the cytochrome it will reach to the photosystem 1, and ATP synthase will have the F0 particle and F1 particle. F0 particle used to make the facilitated diffusion. What is facilitated diffusion? I discussed. And the stroma side, the F1 particle will present in the stroma side. Now I am telling you inside, outside, book also you have written. Now you see how you lost. Out of the following, which are present inside the membrane? Inside the membrane, that is splitting complex and plastocyanin are present inside the membrane. Outside the membrane, what are present? He will ask. Primary electron acceptor of the PS2 and PS1, and next here F1 particle. He will ask. And which are present in the membrane? In the membrane, photosystem 1 and 2, plastophenone, cytochrome B6. These are the questions he can ask. So like that, if you think and it will be, you will create, definitely I think it may come in the next exam. Because the last video also I told, there is no rule how many questions can be given from the which each chapter. And previously, one more thing I am telling you, in the starting, means in the 17, 18, 19, they given more questions in the biotechnology. And next will be in the chapter he has given 1 to 6, 7, but the chapter is less than this chapter. Now he is migrating, he may take this one also. We cannot say be perfect, remember this. If you see my all the videos, it will continue videos, it is there. Video 1, 2, 3, 4, 5, all will cover each and every topic, each and every paragraph. So, subscribe the channel and see all the videos, and it will be from the base to the who are in the 11th class also it will be useful, because they have to give the exam. In this way, if they prepare, it will be useful. So, thank you. If you are feeling good, subscribe my channel, and if you are getting any doubts, please call me, I will give you explanation.